Hello, everyone, and welcome to phyloseminar.org. This is the first talk in a series of 3 talks on the intersection of phylogenetics and cancer research. Please use the YouTube live comment box to ask questions. Today's speaker is Niko Beerenwinkel. Dr. Beerenwinkel is full professor of computational biology at the Department of Biosystem Science and Engineering in the ETH Zurich and Basel. His research is the interface of mathematics, statistics, and computer science with biology and medicine. It includes statistical, evolutionary, and network modeling of molecular profiling data. He has developed several computational methods for supporting molecular diagnostics in oncology and virology, including for the analysis of tumor single-cell genomics data and of deep sequencing data obtained from virus populations. Welcome, Niko, and thank you for participating.
Thank you very much, Eric, for this nice introduction and for the invitation. It's a great pleasure and honor to be here. So I would like to share some of our work on modeling tumor progression from single-cell sequencing data. And I'm going to start right away by reminding us that cancer is, of course, an evolutionary process, a somatic evolutionary process, where cells divide, accumulate mutations, and some of them may confer a selective advantage. And they can give rise to growing tumors, which eventually, may leave their tissue of origin, form carcinomas, and maybe eventually even metastases in different tissues and different organs. And by then, certainly, is usually the time when they become clinically relevant. So there is, of course, a significant clinical outcome with the somatic evolutionary process. And that's probably the main perspective we have on this process. So we would like to describe in some quantitative way the way the tumor grows, and in particular the way the tumor changes genetically, accumulates mutations, and in particular those mutations that are relevant for clinical outcomes. So we do this mostly based on single-cell sequencing data, and I will talk only about single-cell data today, although I want to mention that in general, there also exists approaches to work from bulk sequencing data, and of course other types of measurements for that matter. But the single-cell data is very powerful to approach the so-called tumor phylogeny problem, which consists in, reconstructing the evolutionary history of the cells that form the tumor. So starting from a biopsy of the tumor, which would consist of different cells or different types of tumor cells, genetically different types of tumor cells, we would sample a number of cells and use single-cell sequencing to assess their genetic composition. So in the simplest form, that may look like this binary matrix here, where we have observed a number of mutations in a number of cells, and the matrix would just record for each mutation whether it occurs in a particular cell or not. Then we put a 0. And this little cartoon here already indicates that these data will not be perfect in general. There will be missing data. There will be false positives and false negatives shown in red here, because the data comes from individual cells. So individual molecules have to be amplified in order to get eventually a signal. And that turns out to be a very stochastic, and in the stochastic process that gives rise to quite noisy data. In any case, so that's in a way the mutation data we observe if we restrict to heterozygous mutations, say. And the goal is then to reconstruct a tree, a phylogenetic tree, such that the tips of the tree are labeled with the cells of the tumor. And of course, mutations would occur on the branches of the tree. So that's what we would like to do in the simplest form of the problem. Now, before I talk a little bit more about how to do this and what the challenges are, I want to mention briefly that this is certainly of clinical importance, and I'd like to show these 2 studies, one of which is shown on the left here, which are quite old. So this is a paper from Carlo Meli, 2006, that simply counts the number of different clones that were found in the tumor. And if you have a large number of clones, so if you're in the upper quartile of the distribution of the number of clones, then it turns out that the progression to disease, in this example for a certain adenocarcinoma of the esophagus, is more likely to occur than for a small number of clones. So in other words, the higher the number of clones, the more diversity there is in the tumor, the higher is the risk for disease progression. And this is independent of what the mutation is. It just talks about the diversity within the tumor. The second paper I want to remind us of is a 2015 New England Journal of Medicine paper that actually looks at the order of mutations, in particular mutations in these two genes, TET2 and JAK2. And again, it turns out the order in which they occur determines the risk of progressing in the disease. So here it is for a myeloproliferative neoplasm, and it turns out that the chance to be without thrombosis is much higher in the TET2 first group as compared to the JAK2 first group. So not only does the diversity of the tumors matters, but it matters also in which order the mutations occur. All of the patients here on the right-hand side had both of these mutations, but the order in which occurred made a difference for the clinical outcome. And phylogenetics is one way to try to estimate the order in which mutations occur. And that's a major motivation why we're trying to do this. So I want to mention that we are involved in several different cancer-related projects. A larger one that has been very formative for what we've been doing is the so-called tumor profiler study, which by now has evolved into the Tumor Profiler Center. That is a large effort in Switzerland and mostly in Basel and Zurich, where tumor biopsies have been analyzed with a range of different technologies. Some of them are standard technologies to use in clinical practice, but several of them are more research-grade explorative technologies, where the goal of this study was in the first place to assess feasibility. Can we analyze the same tumor, cells from the same tumor with a range of different technologies? Can we analyze the data, integrate the data, and bring it within 2 weeks into the molecular tumor board, such that within 4 weeks, the data and the analysis results would be available for clinical decision-making and arrive at patient care, actually. So there's a lot to talk about this project, but I just want to mention that our part in this project was just one of these many errors, which is labeled single-cell genomics and single-cell transcriptomics. And I will mostly talk about only single-cell genomics today. But just to put this into perspective, the goal is really to integrate these data and eventually make it useful for decision support. All right, now with the tumor phylogeny problem, there's a number of challenges, and I just want to briefly mention that there are several challenges regarding the inference of these tumor phylogenies. And it's probably a good moment to say that, of course, there is a very large body of literature and methods in phylogenetics, but there's some things about tumors that are, well, special in some sense, at least non-standard. And the first one here is that the genomic events that occur in tumors are not only point mutations, but there's also more complex rearrangements of the genome. Some of them give rise to copy number alterations, some not. So that makes things much more complicated. Data sets tend to be large depending on the technology. We are assessing a large genome, so there can be many mutations. And since we're doing single cells, there may be a large number of cells that we analyze. And I already mentioned that observations are noisy. In particular, false negatives are a major problem. And they would arise from our failure to amplify a mutated allele. And then even if you are able to reconstruct these trees, I think there's maybe even more interesting challenges in what to make out of these trees and how to get interesting information out of them. We might be interested in detecting selection, which mutations, which genes give a selective advantage to the tumor. We might be interested in integrating other single cell observations, as I just mentioned, for the tumor profiler study. We might now have trees for a whole cohort of patients and of tumors. So we want to compare trees. We may want to find subgroups of trees, cluster trees, and so on. And we might want to find things that are common in these cohorts. So we want to find common evolutionary patterns. And I will touch upon some of these points on the top here, and a smaller number of the points from the second part here. All right, I want to start by talking about how to solve the simplest, in a way, tumor phylogeny problem, and then move on to more complex variants of it. I will sometimes work with the so-called cell lineage tree that I've been showing you before, but often I prefer to work with the so-called mutation tree. So in the mutation tree, the vertices of the tree actually correspond to mutations, and the cells can then be attached to that mutation tree. So that is, in a way, an orthogonal representation of the evolutionary history. And sometimes it's advantageous to work with the mutation tree. Sometimes it's advantageous to work with the cell lineage tree. And I will try to make clear when I use which and also why. Okay, so here's the simplest version of approaching the problem. So suppose we have M cells and N mutations. We denote by T the topology of the tree, and sigma is the vector of attachment points for each cell to the mutation tree. So here we're working with the mutation tree, and sigma tells you where to attach each of these cells. So the model has only two continuous parameters, namely a false positive rate and a false negative rate. It is absolutely key to account for noise in the data. And it's also very important that you have two different parameters, so false positives versus false negatives, because false negative rates tend to be very high, maybe up to 20%. Again, they come from failure of amplifying the allele, whereas false positives are much rarer. So the likelihood then simply factorizes over both cells and mutations. So you get one entry for each entry in that binary matrix I showed you earlier. And here you just have the probability of the observed entry of the matrix, Dij, given the expected one. And so the expected one would be given by the topology of the tree. And if you make the infinite sites assumption, which is very common in this context, it just means the mutation can occur at most once and will then be passed on down this tree. And this probability here just accounts for errors. So there's just a confusion matrix here parameterized by our two parameters, alpha and beta. So in a sense, I would argue that's the simplest possible reasonable model for tumor phylogeny, looking only at heterozygous mutations, but accounting for the specific error patterns in the data.
Sorry, Niko, if you don't mind, can you just, I'm a little bit confused about sigma.
Mm-hmm.
So,
okay.
So
sigma
how...
yes, sigma is a vector that tells you for each cell where to attach it to the mutation tree. Right? So if this is cell number 1, then sigma 1 would point to this mutation node. Sigma 2 would point to this one, and so on.
So in a sense, in your likelihood, there's really no edges. We're looking at, we have a bunch of different nodes, and those nodes have certain observations. And what we're looking at is really just the likelihood of the observations given the underlying cell mutations with this error matrix. Is that true?
I mean, this expected value here depends on the topology of the tree. So of course, the topology enters the likelihood, T here, but there is no notion of a branch length here. So we're only accounting for the partial order of the mutations.
Okay, so there's a, if I may just repeat it back. So there's a deterministic function that maps T and sigma to EIJ. So
That's
in a
right.
sense, we can think of, EIJ as a function of T and sigma, and that's how the sigma and the T enter the likelihood function.
That's correct. Absolutely.
Okay.
Yeah.
I appreciate that. Thank you. And while I have you bothered, do you mind, there's a little, it's a notification you're sharing your screen. Do you mind just, don't say stop sharing, just say hide in the bottom
You want me
center.
to move it or?
Yeah, sure. Just like that. That's great. Perfect. Thanks.
All right. Thanks. Okay, great. Now, this vector sigma here is something we are usually not interested in because we don't really care where these cells are attached. What we care about is the partial order of the mutations. Also keep in mind that almost always you don't have additional information about these cells. These cells are destroyed when you extract the DNA for sequencing. So this is different from a classical phylogeny problem. You know, you do human, mouse, dog, and you want to know, right? Are we closer relatives to mice or to dogs and so on? But here it's a cell where we know nothing about. So it doesn't really matter. We're not really caring about the relationship between these cells. What we care about is the process that has given rise to this diversity that we observe. And so what you would typically like to do is to get rid of sigma. So you want to integrate it out or sum over all constellations of this vector sigma. And it turns out that this can be done separately for each cell. So it factors over the cells. And therefore, the computational complexity of this marginalization step is just O and M, where again, N and M are the number of cells and mutations respectively. So in other words, it can be done in time linear in the number of mutations. All right, what you would then still have to do, of course, in order to do inference is find the optimal tree topology, and that remains a hard problem. So we're using a Bayesian approach. So we try to sample from the posterior distribution. And for n mutations, you have n plus 1 to the power of n minus 1 mutation trees. These are just the labeled trees of this size. And in particular, the size of the tree space now does not depend on the number of cells anymore. We still have to marginalize in time that's linear on the number of cells, but then the search space does not depend on the number of cells anymore. So in other words, this formulation and this approach is quite efficient if the number of mutations is moderate, but the number of cells can be very large. So that's why we prefer the mutation tree representation in this particular setting. And then for sampling, I will not talk about how to do this, but the prune and reattach step is, of course, a standard thing that allows you to construct a Markov chain that will converge eventually. And there's some maybe additional tricks how to move in this space, but this is not so different from standard ways of doing it.
Niko, do you mind? Are you going to talk about the prior?
No, I didn't want to talk about the prior, but I can. You mean the prior over the tree space, right?
Yeah, I mean, so you have, you have a prior term in the previous slide.
Yeah, there's no particular assumptions usually about the prior. We try to use a flat prior, and that's really it.
Interesting. Okay, thanks.
All right. Now, next, I want to step back a little bit, and rather than starting with that binary matrix, really the raw data is, of course, sequencing reads. And then maybe after a mapping or alignment step, it's allele counts at specific positions of the genome. So since I mentioned already that the mutation calls tend to be very noisy, a better strategy might be to do the mutation calling and the tree reconstruction at the same time. So now we're starting with the raw data, so the total read count and the counts of the alternative allele. Most positions, of course, have at most 2 different variants, so that's usually good enough. And intuitively, it's of course clear If you have good mutation calls, you will get to more accurate tree reconstructions. But if you think about it, the reverse is also true. If you knew the tree, you would be in a much better position to get the mutation calls correctly in the first place. So the idea is to do now joint inference over the tree and the mutation calls. And we are doing this now. I mean, we started doing this some time back, and let's say the most recent variant of that model also relaxes the infinite sites assumption that so far we have been taken. So now we take into account not all, but several common violations in tumor evolution of the infinite sites assumption. In particular, loss of heterozygosity is a very common event in tumors. So now we allow the loss of the wild-type allele or the loss of a mutated allele in the course of tumor evolution. And we also allow parallel mutations. So we allow the same mutation to occur in parallel in different branches of the tree. So they would occur independently. And as you can see from this representation, now I'm switching to the cell lineage tree, which turns out the better representation for this joint inference task. And so now writing down the likelihood, rather than integrating out the attachment of the cells as we did earlier, now we're integrating here out the placement of the mutations on the edges of the tree. This is maybe more familiar. And again, that can be done for each mutation separately. So this is this factorization over I here. And the probability for each one, for each of these mutations now is modeled, well, by a mixture model, if you wish, because we're considering now all these possible ways in which that mutation can occur. So this accounts, for heterozygous, homozygous, and so on, wild-type loss, mutated loss, accounts for these different possibilities for such a mutation to occur. So that's the model now. And again, of course, due to this factorization, that can be done in time linear on the number of cells. And just to show you very briefly that this is really beneficial, I don't want to discuss this in great detail, but we assessed now two things. One is, of course, the ability to call mutations of this approach. And the other one is the accuracy of tree reconstruction, because again, we're doing both things together. So for the mutation calls, the method is in particular much better than another method that does not take into account the phylogenetic relationship of the cells. So there's a lot of power in doing that. And that is visible here as a function of the number or the fraction of losses that I have in the data, the fraction of parallel mutations. And if you have both of them, that still works very well. And for tree reconstruction, it's similar. You get a better performance when accounting for these events. All right, the next generalization or variation of the problem maybe I want to look at is to look at more complex genetic events. And specifically, we want to model tumor phylogenies now based on copy number alterations, because copy number changes is something that can be assessed quite well even in single cells. So the problem now on a very abstract level looks similar, but in detail is quite different. So the data we start with, at least in the simplest formulation, is just the total read counts. So the cells are here, the rows of this matrix, and after binning the genome, you just count the total number of reads per bin. So this is typically done with low-pass whole genome sequencing data. And so the bins have to be adjusted such that you get a reasonably robust signal per bin. Then from these bin, in the next step, we would detect breakpoints, or maybe you want to call them breakpoint candidates. So these are points in the genome where we might expect the copy number to change. This is done for all cells together. And then we do the actual phylogeny inference. The phylogenetic tree now looks like this. It's a mutation tree type of object, or maybe an event tree, because the vertices are, of course, not single mutations. They are now copy number alterations, and that means we're looking at a consecutive stretch over the genome covering some of these segments between breakpoints. And that stretch can jointly either be amplified or deleted. So plus would be an amplification here, and minus would be a deletion. And so if you trace down a path of that tree and you apply all these events, the copy number profile that you end up with would define the genetic makeup in terms of copy numbers of the cell, or the number of cells, the set of cells attached here, called a clone, for that particular mutational pattern. All right, and from here you can, of course, now predict the copy numbers. They would be in each clone, in each individual cell, they would be integer valued. So they are shown here. So in a way, you can think of this whole process as denoising the raw data into integer copy numbers, but at the same time, we are constructing the full evolutionary history where the events are no more complex genomic rearrangements, or at least the resulting copy number alterations from these events. So that's in a way a much harder problem, because the space of trees, of labeled trees, is much larger. The number of possible events now got, I mean, it's already too large for point mutations, but it became even larger now by looking at these copy number alterations. And again, I want to go quickly and not discuss performance in detail, but in analogy to what I showed you before, we look at the performance in copy number calling. So that would correspond to the mutation calling performance earlier. And this is compared here to a bunch of other methods, and that turns out to work very well. And of course, again, we look at the performance or the ability to reconstruct a tree. And again, on simulated data, that looks very well. So I want to show you two examples briefly. The first one is sort of a data set that has been out there rather early, 2016. It's only 260 cells from a breast cancer xenograft. The raw data is on the top right here. The tree is on the left, of course, and then the predicted, again, integer valued copy numbers are shown here in the middle. You see the tree is quite complex. The events are here labeled with genes that have been affected by the copy number alteration, and then they have been marked with the corresponding copy number change, +2, -1, and so on. Two of these individual cells are highlighted. The first one with this greenish color here on the top, and the second one with this, I guess, brownish color here. Their predicted profiles can be found here, and the individual copy number profile is displayed at the bottom here. So the green one is on the top and the brown one at the bottom. And you get to realize that these data are extremely noisy, and it would be extremely difficult to call these copy numbers for each cell individually. So this is only possible to do with some confidence in the context of the tree. So sharing data according to the evolutionary relationship between cells allows you to make these calls even in the presence of so much noise in the observed read counts. The second example I just want to mention, first of all, this has a much larger number of cells, over 2,000, so the method still works very well for larger numbers. And it turns out that in this tumor, at least we predict a genome duplication that occurred very early on in the evolution of the tumor. So we're starting here with a tetraploid genome, and this can also be detected simply by running the inference method for the situation where you start with a diploid genome versus a tetraploid one, and then comparing the two models. And here we got a clear signal for the tetraploid case. So this is predicted to occur early on here. And in fact, genome doublings are a quite frequent event for several cancer types. All right, the next step, of course, must be to combine copy number changes and point mutation data. And we have done that so far for specifically for panel sequencing data. That's a situation where you have good quality point mutations, and depending on the panel, you can tile the genome and then also have a reasonably well handle on copy number alterations. So that method we call compass, and it now has a component of the likelihood that accounts for copy number alterations, and a component of the likelihood that accounts for point mutations. In particular, this model now also allows for allele-specific mutations and allele-specific copy number alterations. So here's one such example for an AML, a case, acute myeloid leukemia. And just to show you one example, so here we predict two different mutations on the two alleles of TP53. And then after that, a loss of heterozygosity, but it occurs independently and differently in two branches of the tree, such that one allele remains in one branch and the other allele in the other branch. So this is a classical tumor suppressor inactivation mechanism, mutation followed by loss of heterozygosity. But in fact, it's realized slightly differently in two different branches of the tree. So this level of resolution you get to see now with single cell sequencing data and methods like these. All right, the last bit I want to talk about is now switching a bit gears, and now assuming that we can estimate these trees from single cell sequencing data. So I hope I have convinced you that it can be done somehow. And as I mentioned in the beginning, we can ask different questions now on these trees. And if you have a cohort of these trees, then you may want to ask about commonalities or common underlying processes that drive these tumors. So here's a method for inferring the underlying fitness landscape of a tumor, where of course we make the assumption that the tumors are independent realizations of the same, or maybe at least similar evolutionary process. So now we have several patients, and we have a tumor from each of these patients. We did single cell sequencing, and we reconstructed the evolutionary history of each of them. And now we start with these trees, and we try to assess how they have, so what has given rise to these trees that have been observed in these different patients. And again, the assumption is that these are independent observations of the same evolutionary process. This is the type of data and assumption one cannot often make in evolutionary studies. And in that sense, I think tumor evolution is also very exciting, from an evolutionary biology perspective, because even if imperfect, you get to see independent repetitions of the same process. So we like to ask, what's the underlying fitness landscape that gives rise to these trees? So the model that we're using for this is a so-called tree structured branching process. So now we're moving from a phylogenetic model to a population genetics model, a model that has an explicit notion of a population size. And I first want to argue that it's important now to consider population size, and that's for two main reasons. The first one is already for learning this model, it's very important, because after all, we have quite a strong observation bias. I talked about independent observations. But of course, we are seeing these tumors quite late in their progression. We're only seeing them usually when they become clinically relevant. And at that time, the tumors are relatively large. They have at least millions, but maybe more typically billions of cells already. They may even already have formed metastases and so on. So there is an observation bias in the sense that we tend to see the tumor only when it is relatively large already. And it's of course obvious that population size is interesting for predictions that we would like to make, because we're interested in tumors that grow larger. because then they become clinically relevant and become more of a problem. So the branching process, in a way, is very simple. It just accounts for mutation. It, of course, accounts for cell division, for replication, and it accounts for cell death. The way that the cell division rate is modeled is as a function of the fitness landscape. And we're parameterizing the fitness landscape in such a way that it has marginal effects for each mutation that we consider, where we summarize mutations on a per gene level. So for each gene, there's a contribution to fitness, to the growth rate. And we account for pairwise interactions between mutations. So the fitness landscape, again, would be the marginal contributions plus pairwise interactions.
Niko, when I think about this, like a blue dot, so that's a mutation in a quote unquote blue gene. Right?
This one here?
Yeah, for instance. So should I think of that as being like that's a knockout mutation, or that's like some sort of like, I mean, if you had like copy number alteration, that could be an increase of potency of that gene, or how
Yeah,
when you say a mutation, what does that actually mean?
it means, of course, a well-defined fixed mutation, but we're thinking of a somatic mutation that has occurred during the evolution of the tumor.
So, but it's not, I mean, I doubt it's like at position X, base becomes Y. Or is that what we're thinking about?
Yes, it's
Oh,
that.
it is. Okay.
It's
Wow.
that.
Okay.
Exactly.
So it's at that level of resolution. It's not like a mutation in TP53 or something like that.
Yeah, no, sorry. I mean, it's what you said earlier, maybe in the strictest case, but then indeed we often summarize these events on the gene level exactly the way you said. So we would, I mean, for some genes you have to be more careful, but we would summarize usually mutations on the gene level, at least those that we have reason to believe would have the same effect on the gene.
Okay. So this is aggregated across people. So I see. Okay. So there's some aggregation of like these are sort of supposed to be morally equivalent mutations in the sense that they should have the same effect on function. Okay. That's exactly what I was hoping.
You know, this is yeah, but that's in a way, we're talking now about the resolution at which we model mutations. And of course, ideally, you would like to take the exact same genetic alteration, but that is difficult for several reasons. I mean, the data sets are quite limited in size still. Of course, we're now learning something across different tumors. So you have to somehow talk about the same mutation in different individuals. And the exact same base pair change does not occur maybe so often across different patients, but the same gene being hit does occur. So that makes it, possible, or at least easier, to compare across different patients.
Thanks.
All right, so that's the model, but here are a few more details on how to do this. So first of all, we assume that there is a static, a constant wild-type population. So think of a pool of stem cells that give rise to this tissue. Then there is just a way to build trees, let's say, or to grow trees. There's no particular restriction here. You can do it with the infinite science assumption, but you don't have to. It's not important for this model. There is the branching process that I already talked about a little bit, and here is more explicitly the parameterization of the fitness landscape, and I already explained it to you. And we are modeling now the sampling time with a rate that is proportional to the size of the tumor. I mentioned that, and this is how it is implemented. And the last thing here I haven't mentioned yet is that, of course, the sample you're taking from the tumor, well, is a subset of the cells that are actually present in the tumor. So we also need to take into account this sort of subsampling process when we take a biopsy from the tumor. That's the model. And it turns out that these branching processes are very nice and elegant, but can be difficult to solve if a state space becomes a bit more complex, like in this tree structure case. And my PhD student Zhang did a great job solving this by finding this approximation here. So what we really interested is this quantity here called CBT. That's the number of cells of a specific subclone denoted by V at time t. That's what we're trying to model, or that's what the branching process models. And that's what we need in order to assess these probabilities and eventually compute the likelihood. Now that's difficult to assess, and the approximation basically says, well, approximately there is a time-dependent deterministic function. So all the time dependency is approximately just deterministic and in fact very simple. And the stochastic part is in a time-independent random variable denoted here by C tilde. So that approximation is based on earlier work that has been done by Tibor Antal and others. That approximation turns out to be very good. We assess this in simulations here for different shapes of the fitness landscape. These plots here always compare simulation with theory, the solid line with the dashed line. And in all the scenarios that we came up with, the approximation works very well. Formally, it works in large times and small mutation rates, but for the parameter range that is probably relevant here, it turns out to be a very good approximation. So that's the key why this whole model is eventually tractable, numerically even tractable. All right, so I want to show you a few results. So this is a data set of a total of 123 tumors, lymphomas, AML. That's a collaboration with MD Anderson. It was based on panel sequencing data. We further limited it to just 15 genes. So these 15 genes are labeled here. Their mutation rates are also estimated in the model shown with these orange bars. And the marginal fitness effects called base effects here are shown in this column. And then the matrix is shown here. There's an additional regularization term in the likelihood that will drive most, or at least a lot of these entries of the matrix to 0. But whatever survives here is estimated to have a appreciable effect on the growth rate. And so very strong positive effects are in violet, like this one here, for example, and very strong negative effects would be in green in this matrix. So some positive ones are spelled out here. Particular negative interaction was between FLT3 and NRAS mutation, and so on. So in particular, this model now distinguishes, of course, mutation from selection. There's an explicit population genetics notion of a growth rate of a fitness landscape versus mutation rate of these tumors. All right, so now we have sort of a generative model for these trees. And while these fitness values and maybe these interactions are in general of interest, also maybe to find vulnerabilities of tumors and so on, having a generative model, we can also try to ask whether we can do predictions. So that's what's usually called evolutionary forecasting. And there's not much data out there to assess that, but I just show you the little bit we could do so far. So for a few of these AML patients, we actually have 2 time points from the same patient. So here's one such example. The tree is plotted here left to right. Here's the root, left to right. And then the clones at the first time point are all lined up in this column here. And all the clones from the second time point are lined up in the second column here. And then you see there is 3 clones here that have been observed at the second time point, but not at the first one. So the first number here is the relative frequency in percent, and that was just 0. Total number of cells, 0. All right. So in other words, we have here clones observed at a later time point, but not at an earlier. So we can now use the model, of course, excluding this particular patient, and use the data from the earlier time point and try to predict the second time point. What exactly we are using for prediction is a product of these three quantities here: the parent subclone size at sampling, because that obviously affects, let's say, the ability for the tumor to generate that particular clone. So you look at the size of the parent subclone. The mutation rate, of course, should affect that probability, and in particular also the growth rate. So if that mutation occurs, what is the chance that it becomes detectable, maybe even reaches fixation within that period of time? So it's the product of these three quantities that we use as a predictor. And then based on that, maybe I'll show you all of these already, we look at the percentile rank where the observed clone ends up relative to our predictions. And that's shown for our model fit tree in this orange color compared to other models. And then you see, in a way, the further you are to the right, the higher ranked is your prediction. And this is done for these two mutations here. And there were two other patients where we could do the same thing. So there another clone, of course, was involved. And here the prediction worked maybe even a little bit better. So more validation is certainly necessary, but the model now allows you to do these predictions, and as much as evolutionary outcomes can be predicted for these tumors, which depends on how reproducible the process actually is versus stochastic or maybe driven by individual factors of patient's environment and so on. The model provides a framework to do that, and might eventually be useful in designing treatment such that you can account for the evolutionary response that you can expect when applying a certain selective pressure, usually in the form of a therapeutic intervention. So what we're currently doing is we're trying to move this one step forward by also accounting for clinical covariates. So that would allow us to account for some of the differences between patients that probably do play a role. And we also have data from the tumor profiler project that I mentioned very early on single cell drug response effects, and they can be integrated into this framework, such we hopefully eventually arrive at a notion of predicting clone-specific drug response for individual patients. All right, I would like to stop here and maybe briefly sum up. So single-cell tumor phylogenies reconstruct the evolutionary history of a tumor. They may include copy number alterations and/or single nucleotide variants. They can serve as the backbone for integrating and denoising single-cell multi-omics data. I did not talk about this very much, but we're working a lot on integrating RNA sequencing data, proteomics data. Well, I at least mentioned drug response data in the end, but I didn't really show you how to do it. They can help identifying evolutionary biomarkers. By that, I mean features of the evolutionary process that are predictive of treatment outcome. They can help identifying patient subgroups with similar tumor evolutionary patterns. That can be done with the model I just showed you, but there's also more direct ways that we and others pursue by simply clustering trees in different ways and finding groups of patients that have similar tumor phylogenies. And finally, the single-cell tumor phylogenies can predict future evolutionary events. And I think this is a very exciting direction to go, and I hope will eventually be clinically useful. I want to stop here and thank my group for doing this exciting work with me, and our collaborators, alumni, and the funding agencies. And thank you for listening.
Thank you, Niko. That was really exciting. I especially love the last part where you were integrating fitness in. And also just the fact that you do get just sort of on the ground use out of phylogenies by improving the calls. And that, I mean, I think that that's always been a vision that like by getting better phylogenies, you kind of get better data cleaning and so on. And then that's actually working here. So that's exciting. Actually, a lot of the questions that I had came up in your sort of future things. So can you say a little bit about how you're thinking about integrating the gene expression data or how you're doing that?
Yes, sure. So for the gene expression data, it's in a way relatively straightforward, although in detail, yeah, depending on how you do it, it can be complicated. It's relatively straightforward in the sense that if you have from the DNA a copy number signal, so if you do a whole genome sequencing, or the panel is tiling the genome and so on. So if you have a reasonably good copy number signal from the DNA, you can also get a copy number signal from the RNA, because, very roughly, the RNA is proportional to the DNA. The more DNA, the more RNA is transcribed, or the more DNA is transcribed into RNA. And so the models trying to do that are, of course, all based on that basic idea. You typically have to go to larger genomic regions, because on the RNA level, of course, there's other processes operating, gene regulation after all. But over large genomic regions, that signal becomes clearer and clearer. So that allows you to map single cell RNA data onto single cell DNA data, or onto the tree derived from single cell DNA data. Maybe I should, to be clear, I should say that the typical situation is, of course, we have a tumor biopsy and we split that biopsy, maybe after dissolving a solid tumor. But from the cell suspension, we take part of it for single cell DNA sequencing, part of it for single cell RNA sequencing. So in other words, the individual cells are different, they're not matched, and hence we need to solve this mapping problem of the transcriptional diversity onto the genetic diversity. And again, the copy number signal is quite useful for that, of course. So that works very well. And then it depends what other measurements you have. So for the drug response data, for example, that is much more complicated. I mean, we work with data that Behrend Snyder generated. It's called pharmacoscopy. These are single cell imaging based readouts. And there's no such direct, obvious way to relate such an image-based single cell readout to copy number alterations. And then you have to try different things. Of course, one thing is the whole structure of the tumor, the size of the clones, the composition. But then you can also try to look at, let's say, pathway level alterations. So the image-based readout will have some markers that may give you a clue which signaling pathways or other pathways are affected in the tumor. The same can be done for copy number data, and that may give you some additional clue on how to match these two. But that's already a much more difficult task. Proteomics data, it depends what exactly it is. It's individual markers, so they can maybe relate more directly. But overall, it's a form of distribution matching that you have to do in one or another way.
Wow. Yeah. Thank you for explaining that, because that was not obvious to me. I mean, I guess now that I think about it, of course, you can't do sequencing and these other assays on the same cells, but that does make it harder. So we do have two questions, both from Amit Prakash. So they ask, could there be a way to predict chemotherapy or cancer therapy in general using tumor phylogeny predictions?
Yeah, that's a tough question. I would say that this is certainly a long-term goal we have, but the phylogeny alone doesn't, of course, directly tell you whether the patient will respond to the treatment or not. So the way I see it is that the phylogeny is a very useful ingredient in doing that. And it's also a very good sort of backbone for integrating other single cell measurements. But the phylogeny alone will probably not answer that question. I mean, it could be, but it seems unlikely to me that we will find the magic tree feature that tells you whether patients respond or not. But again, I think it's a very natural way to integrate single cell data on the trees, because in the way the genetics, even if they don't in the end determine response completely, in a way you always need to take into account the genetics properly, because they affect any other molecular constellation of the cell, of course, RNA, proteins, and so on. And so it's, in a way, I think it's not an option, or at least not a good way to go about to ignore the genetics. And therefore, it seems to me very natural to start with it. It serves also to denoise any other molecular measurement, right? If there's a transcriptional gene expression change, for example, you would like to know, is it driven by a copy number alteration, or is it driven by an environmental cue that will have consequences how you interpret it, and what it may mean to the fitness and the response to treatment of the tumor.
But just to dig a little deeper, I mean, I agree that you shouldn't be able to just predict it from the tree alone. But I mean, you presumably, the people, when you're doing these prediction exercises, these people are getting treatment. So, right? I mean, between the different time points, or are they not?
Yes. I mean, I guess it depends, but for the data we have, they typically were treated in between. Yes.
So, I mean, it seems like that's maybe the training data for such a predictive algorithm. You have a bunch of observations. I mean, I
Yeah.
guess it's not quite like, maybe it's not suitable for some sort of causal analysis, but yeah, it seems like you could do something there.
Yeah.
But that's interesting.
Absolutely.
So, I mean, also asks, where do you see this work heading in the future? Where's the room for improvement? You've talked on some of those points. Is there anything else you wanted to mention?
Yeah, I think there are many. I mentioned some of them. I think this remains a very difficult problem. Of course, I showed you how we approach it, but neither our nor other methods are perfect. So I think there's quite some room to do this better. It also depends quite a bit on what exactly the data is. You can do a single cell panel sequencing. You can do whole genome sequencing. You can, of course, do exome sequencing, and then it depends what exactly your amplification method is, what exactly your panel is, and so on. All of these things make a difference. Then you may have time series data or not. You may have multiple biopsies from the same tumor. All of this has an impact. So I think there's a lot of things to improve. The genetic alterations in the tumor tend to be complex, and all I showed you in this model was accounting for copy number alterations, but really there is a specific genomic rearrangement underlying, right? And we are not keeping, in this model, we're not keeping track of what exactly happened. So if there was a amplification, for example, we don't keep track on where these copies of that genomic region ended up in the tumor genome. So in other words, there is a level of modeling it more precisely by accounting for the breakpoints and keeping track of all parts of the genome where they go if duplicated, and so on. And there's also copy number neutral alterations, like a translocation, that would not even be captured with this approach. So I think there's a lot to improve in accuracy. And I think for the downstream analysis, there's just a huge number of things one can ask now. I mentioned a few on my slide. That was a selection of things we like to think about, but I think there's probably many more questions. Comparing trees has almost evolved as a little subfield, because that's also algorithmically a very interesting and challenging problem comparing labeled trees. But then the label sets can be different in this problem here, because different mutations occur in different patients. I think that's a very exciting problem. And so on. I think there's a lot to do.
That's great. Well, what a great note to end on. I mean, a lot of it does seem to come back to this question of like, when do we consider mutations to be the same? I mean, not a lot of it, but I mean, that seems to be a key question here, where you're like, if you want to be able to aggregate information between different individuals and different scenarios of cancer progression, but when do we consider two translocations to be the same? I mean, if you consider everything to be completely independent, it feels like you're never going to be able to make anything generalizable.
Yes, I agree. If you stick to the most precise definition there, then we will probably never have enough data, right? I mean, conceptually it remains the same, but how many patients do we need to sequence until we see the exact same point mutation often enough to say anything about it? Therefore, I agree. Yes, we have to summarize it, and indeed, it's not obvious what the optimal way, in some sense, to do that is. The gene level is kind of a natural thing to do, but I'm not saying it's always the best way to do it. So yes, that's an open problem. Absolutely.
Cool. Well, thank you so much. What a wonderful way to start off our series. I really appreciate it, Niko.
Thank you.
All right. Bye now.